Hello friends, welcome to this video of uronic acid pathway, which is also a minor pathway of glucose oxidation like we did HMP in the previous video. So let's get started. Uronic acid pathway has some similarities with HMP. Let's note down that first. Similarities with HMP. One is that this is also a minor oxidation pathway for glucose. Then this pathway also occurs in cytoplasm. The main organ is liver. The starting material is glucose 6-phosphate and no ATP is produced in this pathway. Okay, so let's start with the pathway. The starting material is glucose 6-phosphate, which will be converted to UDP glucose. This thing, that how glucose 6-phosphate is converted to UDP glucose, this we have already done in glycogen synthesis. Okay, there are just 2 steps. You can see those steps from that video. Now UDP glucose is formed and now oxidation will occur at C6 position, making UDP glucuronic acid. You know that oxidation at C6 makes uronic acids. Okay, this glucuronic acid, UDP glucuronic acid, is used at various places. It is used for conjugation reactions of phase 2 xenobiotic reactions, also in the conjugation of bilirubin in liver. It is used for GAG synthesis, right? Then UDP glucuronic acid is converted to D-glucuronic acid. Here UDP is removed, UDP group is removed by hydrolysis. Okay, so D-glucuronic acid is formed. Then by reduction of D-glucuronic acid, we get L-gluconate. All these compounds which we are talking here, glucuronic acid, D-glucuronic acid, UDP glucuronic acid, L-gluconate, all these are 6-carbon compounds. Okay. Now this L-gluconate is used in the synthesis of vitamin C, or you can say ascorbic acid. This synthesis of vitamin C cannot occur in humans due to the deficiency of the enzyme required here. Which enzyme is required here is very, very important. What is enzyme? L-gulonolactone oxidase. Okay, so this enzyme is not present in humans. That's why vitamin C is needed in diet and it cannot be formed in body. Okay, so till this point all are 6-carbon compounds. Okay, now in next step, one carbon dioxide will be removed and we get L-xylulose. L-xylulose is now a 5-carbon compound. Okay, this L-xylulose is converted to xylitol by xylulose reductase enzyme. Then xylitol dehydrogenase. Next is xylitol dehydrogenase converts xylitol to D-xylulose. So all these are 5-carbon compounds. Now defect in this enzyme or this enzyme is leading to a condition known as essential pentose urea. What do you mean by this? Pentose is coming in urine. So if xylitol dehydrogenase is deficient, xylitol will accumulate. That is also a 5-carbon compound. Or if xylulose reductase is deficient, then L-xylulose will accumulate. That is also a 5-carbon compound. So either this compound or this compound is accumulated in body and coming in urine. So the condition will be known as pentosuria. Okay, so it is essential pentosuria. We'll be doing detail later. Now let's continue with the pathway. This D-xylulose which is formed, it gets converted to xylulose 5-phosphate, and this is one of the intermediate of HMP pathway, so it will enter HMP pathway, right? So the pathway is done. Let's see the uses or functions of uronic acid pathway. Why uronic acid pathway occurs in cells? Why it occurs? Because it will synthesize, this pathway synthesize 3 things. One is it synthesizes vitamin C, but this synthesis does not occur in humans. Then it will synthesize glucuronic acid. I told you this glucuronic acid is required at so many places in the body. Then it is also making pentoses. So these are the 3 uses of uronic acid pathway. Now let's do this disease, essential pentosuria. Pentoses are coming in urine, and this is a benign condition. It is a benign condition, but it should be differentiated from diabetes. It should be differentiated from diabetes, as in this condition also Benedict's test will be positive. Benedict's test will be positive in the urine of the patient. Why Benedict's test is positive here? Because pentoses are present in urine. Pentoses are monosaccharide, and any monosaccharide gives Benedict's positive. So that's why. Okay. But in this case of essential pentosuria, glucose strips test will be negative, indicating that this monosaccharide which is giving Benedict's positive is not glucose but some other thing, and that is xylitol. So which enzyme is defective here? Either it is xylulose reductase which may be deficient or defective, or it is xylitol dehydrogenase, right? And which substrate will accumulate here? It is L-xylulose or xylitol. Both are 5-carbon compound, that is pentoses. This condition, that is pentosuria, can also occur in a normal situation. Like in a normal person, if there is large amount of ingestion of fruits rich in pentoses, then also pentosuria can occur, right? And this condition, essential pentosuria, is a part of Garrod's tetrad. What is Garrod's tetrad? There was a scientist named Garrod who discovered 4 inborn errors of metabolism and grouped them together with his name, that is Garrod's tetrad. Which are these? I'll help you learn with the mnemonic. The mnemonic is CAP. C-A-A-P, CAP. C stands for cysteinuria. 1A stands for L-captonuria. Another A stands for albinism, and this P stands for the condition which we are doing just now, pentosuria or essential pentosuria, right? This is important. You have to learn all 4 diseases of Garrod's tetrad. And this is it with the video of uronic acid pathway. Thanks for watching. Best wishes to you.